The unique storage organelles of the seed cells include protein storage vacuoles, you see here, protein storage vacuoles, PSV, and endoplasmic reticulum derived protein bodies, PBs, and starch granules, you have S, and all of which have been used for protein accumulation in the plant system. Finally, proteins can be produced in roots and secreted to rhizosphere. This strategy is often preferred for phytoremediation purposes. Here the different production system, the platform is given. It's a comparison between various hosts in molecular farming, production of high value biomolecules. Here you have the good systems like fruits, and then the animal, and then transgenic animal, bacterial system, and yeast system. You have the many production system are readily available for production of high value biomolecules. Here you are given the different colors in the chart. Each color indicates different points, advantage and disadvantage, and then the preference of the, you can choose any platform for production of any particular protein. Here you can see the fruits and the nuts are used for production of, some of them are used, plant tissue are used for production of the biomolecules. Here this shows that this expense cost, overall cost is medium level, it is indicated, and then scale up capacity is medium moderate, and then production protein folding accuracy is very high in the plant cells, and then product quality is very high in the plant system, and storage cost is very moderate, and then safety is very moderate, and then public perception of risk, this is very high. Only the drawback with all the transgenic product in plant, especially the plant transgenic product, public perception is high risk. Public perceives something, and then they make a lot of agitations, and then they make a lot of it is all because of the wrong perception. And then you have the transgenic plants, especially the fruits like banana. You see the overall cost very cheap when you target a protein to directly to the banana, and then the banana is we can eat it raw format, we can so directly it will be consumed by us, so that it will be the cheapest overall cost in the production aspect, cheapest source banana. And then storage is much very cheap. You can store it, this banana, wherever you want to store it, and then the scale up capacity is very high, and then production cost is low, and then protein folding accuracy is good, and product quality is very high, protein yield also very high, and the safety is very high. And then again the only the risk factor, the public perception of risk is also very high, which is of drawback in this plant system itself, not this particularly with the banana. Here is you have the another plant virus system. You have again we can say some of them for protein production of some of the proteins. This plant virus system is cheap. Likewise, it depends on the protein, and then the host plant, and then the target product, and then target group for commercialization of this product. We can select any plant system which gives higher yield. And then most of the things like plant cells and the plant viruses, these are the very cheap method, cheap technique to produce high quantity of the product. Here you can see, suppose in case if you take the yeast system, this almost the production overall cost is moderate, only production cost is moderate, and then all of them are moderate, and then the safety is not at all known. Safety is unknown when we produce yeast, but people accept this thing. When we come to the bacterial system, the protein folding accuracy is low. These are the disadvantage in the bacterial system because of this prokaryotic in nature. And then the product quality is low, and then the safety is low, and then public perception it shows very low, because they lack of awareness of this particular production system. So there is public perception of risk also low in this category. Some of the most common plant host systems used for this plant molecular farming is shown here. As usual, the model plant comes first, that is Arabidopsis thaliana. We can generate lot of mutants, and then lot of mutants were already developed using in Arabidopsis thaliana because of the small genome, and then the genetic resource of the genome sequence or the gene characteristics are available in the public domains. And the small limitation is that low biomass production capacity of this Arabidopsis thaliana. So when the plant is low in biomass production, so we need to have the large area to produce to get the high biomass. So that is one of the limitation. And then leafy crops like tobacco gives a very big leaf, so that is a high biomass, and then we can scale up rapidly. And then this is a non-food crop, it's a feed crop. And then the limitation with this tobacco is low protein stability, presence of toxic alkaloids in the plant system. In the alfalfa, the high biomass yield, alfalfa gives very high biomass, and then harvest up to 6 times a year. Every 2 months, once in 2 months, you can harvest tons of high biomass of alfalfa. And then bulk antibody production very high. We can produce bulk antibody in this case. And then only the small disadvantage using this alpha is low protein stability. So here we need to produce or target this, we need to choose this alfalfa for production of the protein which is having the high stability only. So that is the way how we can decide select a particular plant system for production of good protein with low cost of production. Then only we can give supply to the commercial firm with a reasonable price. And then lettuce can take as a edible part, we can eat this lettuce as edible part. Again, this low protein stability, so we need to select a high quality, stable protein to express in the lettuce as alfalfa. And then some of the cereal seed crops, as we have seen in the previous slides, the seed has many compartments in it. So these are the crops like maize, rice and wheat. Advantage is high protein stability during storage, high yield, we can get a huge high yield crops, easy transformation and scalability. Already the transformation, genetic transformation protocols are established in maize, rice and wheat crop. And also the other crops, some of the crops fixes atmospheric nitrogen, that also gives the nitrogen to supply to the plant, and then it leads to good protein production. And then only the difficulty is the downstreaming process with the seed as a source for production of high value molecules. And then legume seeds, the pea and soybean, they are mostly widely used for production of high value proteins. It gives high protein quality, high protein stability during storage, high yield, easy transformation. And then the pulse crops fixes atmospheric nitrogen in symbiotic relationship with rhizobium and other related genera. And then only again the seed comes, the difficulty comes with the downstreaming process when seeds are used as a sink for expression of protein. And then coming to fruits and vegetables, and then especially the potato, tomato and banana, we can, with the fruits and vegetables, potato we can edible, it's edible, bulk antibody can be produced. And then potato is a huge biomass yield crop. And then only the drawback is cooking or boiling. During cooking or boiling degrade the protein. We cannot eat potato or raw potatoes, so we need to cook or boil. So that boiling and cooking leads to degradation of the protein. Tomato, this is high biomass, we know. It gives tons of tomato from acre, and then it is edible. Only to produce these proteins in the tomato, we require a greenhouse, and then low protein stability. Some of the proteins are very low in tomato, and also the tomato is acidic in nature, so we need to have a protein which is stable in acidic conditions. This kind of the points we need to consider before selecting a protein to be targeted in a particular plant platform. And then banana, it's edible, less costly. And then again another low protein stability is a major factor here. And then fiber and oilseed crops also used for production of high value biomolecule proteins. Example, flax, cotton, safflower are the crops which are used for expression of proteins. This advantage is oleosin fusing protein can be produced here, and the drawback is fiber and oil can interfere with downstream processing. Downstreaming process, we have difficulties with the presence of oil and fiber in these crops. And cash crop, sugarcane, it's one of the ideal crops. High sugar leads to stability of the protein first of all, because we are expressing that protein particularly to the vacuole where the sugar is stored. So when we express the protein to the sugar ambience, that naturally gives stability to the protein. And then the sugarcane accumulates highest biomass. And then we have the well developed protocol for genetic transformation, and also scalability is high. The drawback is downstreaming process, because as soon as we extract the juice from the plant, it gives both sugar and protein. It needs to be separated in an ideal environment. Separation is easy, only we need to have some of the ideal environment to separate both of them. It takes time. Then comparison of expression systems. Here I have shown in this table the advantages of and disadvantages or requirement of the different platforms for production of high value molecules. Here we have the 5 major expressions. Six major expression system: yeast, bacteria, plant cell culture, transgenic plants, transgenic animal cell culture. And then the point we consider is cost of maintaining it, and the type of storage, any special storage is required, and the gene or protein, size of the protein, and then production cost and protein yield. When it comes to transgenic plants, the cost of maintenance inexpensive, because you can plant it in open environment or transgenic or any glasshouse or polyhouse. So that is very easy and then inexpensive. And then type of storage not required for plant crop, you need not required. And then gene or protein size, there is no limit in any protein, any size can be expressed in the transgenic plants. And then production cost, the cost of cultivation is low, we can have the production cost also low. And then protein yield, because of protein yield is very high because of high biomass production capacity of the plant system. So this is ideal plant system for production of many proteins. However, some of the proteins require some specific ambience, so that can be expressed, that can be produced either yeast, bacteria, or plant tissue culture, or transgenic animals, or animal cell cultures. Those are the platforms can be used for some specific protein production of some specific proteins. Why plants for molecular farming? Plants had the potential to produce, plant has the potential to produce complex mammalian proteins of the medical importance. We can produce any complex proteins of medical importance in the plant system. And the low cost of production, stability, storage by freezing or drying of leaves, it's simple, and safety, free from any animal or human virus contamination. And then Arabidopsis thaliana is a model plant for production, and then easily reproducible in consumable form. Likewise, cereals, if you express some of the proteins to the leafy crops, the leaf can be consumed as a easily edible part. And fruits and vegetables like tomato, mango, banana, these are some of the examples, easily reproducible. These are the product example of plant molecular farming products in different types of plants. You see this figure shows clearly the different plant molecular farming products in different types of plants. What are the plants are ideal for production of different products. Here you can see some of the example. Vaccines can be produced in hepatitis B antigen, that is the lettuce and potato, and then fusion proteins including epitopes from rabies and spinach, Vibrio cholerae in potato. Likewise, we have a cancer vaccine in tobacco, antigen in tobacco, human growth hormone in many plants, poultry vaccine in canola, the capsid protein normal virus potato and tomato, H5N1 vaccine candidate in tobacco. These are the vaccine category. We have different system like tobacco, potato, maize, spinach. These are the different, tomato also comes, and canola, even canola comes. These are the different plant production platform for production of high value molecules. Nutraceuticals, you have the safflower, and then you have the rice plant. Which can be used for human lactoferrin production, and then human intrinsic factor in Arabidopsis plants, and then barley also used for production of nutraceuticals. Therapeutic human proteins plant systems like rice, tobacco, safflower, duckweed and carrot. These are different platform for production of therapeutic human proteins. For example, insulin is produced in safflower. These are the different plant system used for production of these therapeutic human proteins. And galactosidase in tobacco is another example. The antibodies, you have multiple plant systems, and then multiple antibodies are produced in tobacco. You see almost all the antibodies are produced in tobacco. In almost you see more than 10 antibodies are different related to that group is produced in tobacco plant. These are the simple form of expressing or giving the idea what are the products can be produced from the plant system, and then the biomass related to that, and downstreaming, and then safety, and then speed, and all different advantages are given based on overall production of plant-based expression. Here, these are the comparison of waste stream generated in mammalian cell culture based process relying on single-use equipment and a plant-based counterpart. As we have seen that in the previous slide, this production of value biomolecules in the plant system is a green biotechnology, because it is not leaving any unused biomass in the environment. But whereas when we use these other systems like other commercial system like bacteria and yeast and all, it is going to be added in the environment. That creates a little bit of environmental concern issues. So these are the So whatever we produce in the plant system, whole thing is biodegradable. Whole plant system itself we can it can be degraded. But when we compare to other system, but and moreover this is not contaminated by it will not be affected by the contamination. When we use the bacteria as a production system, if some other bacteria is contaminating or fungal is contaminating or any other fast growing anti-active myces are contaminated, it's a serious issue, serious problem. It creates a lot of loss to the company, lot of health issues to the publics. But when we use plant as a production platform, that kind of concern is not there here. The compiled table of survey of molecular farming commercial landscape from 2005 to 2020 is compiled, and in detail it is given in publications by Fisher and Buell, published in 2020. These are the downstreaming simplistic the method for doing the harvesting and the production of different molecules in the plant system. And then for the success of this plant molecular farming, it depends on the genetic engineering protocol available for a plant system. This involves isolation and procurement of gene of interest. First of all, you need to have which gene you want to express in the plant system. You should isolate, or either you should isolate or procure the gene of interest from a commercial firm, and then this gene needs to be optimized. Its codon needs to be optimized for its better expression in the plant system. And then after codon optimization and the synthesis of the gene of your interest, you need to construct the gene plant expression vector with the gene. And then we need to perform the plant transformation, and then the molecular characterization of the transgenic events, and then western northern blot analysis for further confirmation, and then taking further to production. This is one of the examples of the plant expression vector. Here I have just given different plant-based expression systems are available. One is stable nuclear transformation. This is the most common method of transformation used for transforming the gene of interest into the plant. And then foreign genes are transferred either via agrobacterial tumefacients or the particle bombardments. Genes are taken up and incorporated in a stable manner. Large acres can be utilized with the low-cost grains. Long-term non-refrigerated storage of the seed up to 2 years is possible. Manual rubber is required, only manual rubber is required. Lower yield and outcrossing. Some of them are and then another transformation is the plastic transformation. As we have seen in the previous slide, it depends on the nature of the protein and the requirement. You can specifically target the transform the gene of interest indirectly into the chloroplast. This is depends on your requirement. So it is also another system. And then transient expression, transient transformation, that is most widely used for production of plant recombinant viruses to infect host plant like TMB, CAMB and PVX, occur infiltration through recombinant agrobacterium tumefacients. Small amounts target protein is obtained in a weeks. Infection process is rapid. Protein accumulating in interstitial spaces, because this kind of transient expression system is preferred when small amount of target protein is required within a short period. So that kind of proteins can be targeted as a transient expression. From that infection, based on the infection process, we can rapidly isolate the protein accumulated in the interstitial spaces. Target protein is temporarily expressed in the plant. No long-term storage due to tissue damage. No stable transgenic plants are generated here. We need not to maintain it. These transient transformation events, and the low scalability in the expression level. low stability and expression levels is observed in the transient transformation. The stable transformation for hydroponics. Transgenic plants are grown on hydroponic medium, and desired products are released as a part of the root fluid into a hydroponic medium. Plants are contained in a greenhouse, so the transgenic events can be contained in the greenhouse. From that greenhouse condition, the hydroponics, the plants will be grown on the hydroponics, and then the proteins are circulated in the root system, and then from that hydroponic solution we can easily purify the protein, which is of less expensive, and then this is not suitable for large scale production because hydroponic itself expensive, and then but it is highly suitable for small scale production.